Hi friends, thank you for joining me for another episode of Catch This. Today's episode is not about SARS-CoV-2, but please don't leave just yet. Stay with me. I promise you this will be worth your time. Did you know that our cells and bacterial cells make antiviral agents? Like the same antiviral agents? Stay tuned. Antiviral agents are a class of drugs that inhibit the replication of viruses at various stages of their replication cycle. You may have heard of some of these in the news recently. There's remdesivir for treating SARS-CoV-2 infection, ribavirin for treating RSV infection, acyclovir for treating herpes virus infection. As you could probably tell, they all have the letters VIR in the name. When you hear virin or vir at the end of a drug name, you can probably assume it's an antiviral. One class of antivirals consists of small molecules known as chain terminators, because their addition to the end of a chain of DNA or RNA prevents the addition of any subsequent nucleotides, which are the building blocks of DNA and RNA. And if you can't add any more nucleotides to a growing chain, then this ends the replication of DNA or RNA. Let me explain. Individual RNA nucleotides consist of a ribose sugar connected to one of 4 different bases and to a group of 3 phosphates. The carbon atoms in the ribose sugar, those are represented by the unlabeled corners in the pentagon. If there's nothing there, you just assume it's a carbon. Those carbons are numbered, and the 5th one is actually outside of the cyclic structure. The base is connected to the ribose via its 1' carbon, and this base can be either an adenine, guanine, cytosine, or uracil in RNA. In DNA, the uracil would be replaced by a thymine. The 3 phosphates, also referred to as a triphosphate group, are connected to the ribose at its 5' carbon. For example, the ribose in the nucleotide cytidine triphosphate, also known as CTP, is connected to a triphosphate group and a cytosine base. Addition of this CTP to the end of a replicating RNA strand results in loss of 2 of its phosphates and connection of the remaining phosphate to the 3' hydroxyl of the ribose in the preceding nucleotide in the strand. If a ribose doesn't have that hydroxyl at its 3' carbon, then this would prevent the binding of a subsequent nucleotide and would thus make that nucleotide the last one in the chain. But what does all of this have to do with viruses? Viral infection in humans induces production of interferon. Interferon is a cytokine that further stimulates the expression of other genes, one of which is Viprin. Viprin stands for Virus Inhibitory Protein Endoplasmic Reticulum Associated Interferon Inducible. Research in recent years has shown that Viprin catalyzes the conversion of CTP to 3' deoxy, 3' 4' didehydrocytidine triphosphate. I won't be saying that again, so let's just call it DDH-CTP. As the deoxy portion of its name implies, DDH-CTP lacks a hydroxyl at the 3' carbon of its ribose sugar, and this makes it a chain terminator. In human cells, the conversion of CTP to DDH-CTP by Viprin leads to broad antiviral activity against various viruses, including human cytomegaloviruses, West Nile virus, dengue virus, hepatitis C virus, and HIV. And it has no toxic effects on host DNA replication, like the replication of our own DNA. Some bacteria and archaea encode genes that have notable sequence similarity to vertebrate viprins. However, until recently, their role was unclear. Because of the antiviral role of viprins in human cells, and this was found out in 2018, the authors of a recent publication wanted to determine whether prokaryotic Viprints participate in defenses against viruses that infect bacteria. And as you may well know, these viruses are called bacteriophages, or simply phages. Knowing that genes encoding antiviral defense proteins tend to cluster together in the prokaryotic genome, the authors searched for the presence of such clusters in a variety of bacterial and archaeal genomes. They found that although most clusters of Viprin-like genes did not co-localize with defense genes, 60% of the genes in one cluster were located near genes encoding CRISPR-Cas systems, restriction enzymes, and other bacterial defense genes. To see whether the prokaryotic Viprin-like genes in these clusters encoded proteins with antiviral activity, let's just call these prokaryotic Viprints PVIPs for short. The authors cloned 59 of these PVIP genes into E. coli under the control of an inducible promoter. This allowed for expression of these genes to be switched on or off. When they infected these PVIP-producing E. coli with a range of phages, they found out that about half of the tested PVIPs had inhibitory activity against the phages. Most of the PVIPs inhibited bacteriophage T7, but some of them also inhibited other phages, without any toxic effects on the bacterial host cells. What was surprising was that when they expressed human Viprin in E. coli under the same conditions, it protected against infection by T7 phages. Phages that don't cause disease in humans, so there's no reason why we would have a defense mechanism against them. But this suggests that the antiviral functions of Viprin and Viprin-like gene products are highly conserved between different organisms. The authors next wanted to determine whether PVIPs catalyze production of DDH-CTP, like human Viprins do. If these modified nucleotides are made in PVIP-producing E. coli, having antiphage activity, they would probably be found in small molecule fragments extracted from these E. coli. So the authors analyzed E. coli lysates by mass spectrometry. Mass spectrometry is a technique that reveals the chemical identity of molecules and other chemical compounds. They then compared the small molecules identified from cells producing PVIPs to molecules obtained from E. coli containing human Viprin. This analysis revealed that cells producing some prokaryotic Viprin-like molecules contained derivatives of CTP, including DDH-CTP, while others did not, suggesting that only some Viprins catalyze the production of DDH-CTP. However, some prokaryotic Viprins seem to produce modified DDH derivatives of other nucleotides like GTP and UTP. Collectively, these results suggest that prokaryotic Viprins produce antiviral nucleotides just like human Viprins do. Although environmental bacteria have long been used to identify new antibiotics for treating infections caused by bacteria, the notion of extracting antivirals from bacteria is new. Now, many of the antiviral drugs used in clinical settings are chain terminators, including acyclovir for herpes virus infection, AZT for HIV infection, and sofosbuvir for treating hepatitis C virus infections. So to me, at least, it is really interesting that the present study may have revealed an additional source of diverse antiviral molecules that could be adapted for clinical treatment of human viral infections. What is the biggest take-home message here? To me, the biggest take-home message is that it is so important to pay attention to all science, not just the little niche you may have carved out for yourself. So there may be a lesson in here for me. If you want to learn about the study in more detail, I recommend listening to This Week in Microbiology episode 233. That's it for today. Thanks again for listening. My name is Trudy Ray. Please subscribe. I'll see you next time on Catch This.